I'll share my screen. No, didn't want to. Okay, so I'm finishing up HPLC today, and just to keep you in the subject and refresh your memory, let's go over some recap questions. All right, so how does temperature impact resolution? You all did the lab already in HPLC, and I showed you the oven, and I thought that we will, and I mentioned that we have the temperature at 38. So Dan said higher temperature will enhance the resolution. Yes. How does it do that? Basically, at higher temperature, your mobile phase will be less dense, so the viscosity will be lower, so that will enhance the mobility, as well as your components will have higher kinetic energy, so they move in and out of the stationary phase and the mobile phase, reach equilibrium faster, so you have sharper peak and less diffusion within the column, so your peak won't be a wide. So sharper peak that will enhance separation and prevent coelution with another component that might come out close to that other one. So how does flow rate impact resolution? If the flow rate is high versus low. Anybody remember that? Okay, let's see. So N would say higher flow rate, less resolution, peaks closer together. Flow rate is high and less resolution. Okay, well, what about if the flow rate is very slow? Well, that's correct what you're saying. That a very high flow rate, yes, things will move very fast and you might not get enough time to separate. But on the other end, if the flow is very slow, what do you think might happen, Kat? Yes, so when the flow rate is slow, your components have more time on the column to diffuse and move down the column by diffusion rather than going in and out and equilibrating. So we don't want very slow flow rate, we don't want very high flow rate. So we really need to find the optimum flow rate that gives me the lowest height equivalent of a theoretical plate. So the flow rate plays a very important role, and it really, you don't want it high, you don't want it low, you want to find the sweet spot. How does the packing and thickness of the stationary phase impact resolution? So if we have a packed column versus a capillary column. So in a capillary column, you have a hollow tube and a very thin stationary phase around in the interior. So if you have thin stationary phase versus thick and packed stationary phase, how do you think that will impact the resolution? Think about it. So if you have less stationary phase, do you have better resolution or not? Steve? The thicker the stationary phase, Steve is saying the more the resolution. I would say the thicker it is, the more the capacity. So you would have more capacity, but you might have more space and more diffusion going on. So the thinner the stationary phase, you don't have much diffusion. The component is going to go in and not diffuse, not linger, comes out quicker into the mobile phase. So you really have, with thinner stationary phase, you really have better resolution, but less capacity. So you have to think based on what you really want. You want more capacity or you want better resolution? How much sample you're injecting? Do you want to be able to inject more samples? So you probably need more capacity. So really it depends on your objective. What do you really want to do? But the thickness, so the thinner it is, the better the resolution. That's what you want to remember. What are the advantages of a narrower diameter of a column? So if you have narrow versus wide and big. Say that again, Abby. Flow rate doesn't have to do, is independent of your column. You control the flow rate with the pump. Definitely the column impact the pressure, so sometimes you have limitation on how much you can increase the flow rate based on the pressure generated. But if we want to think about resolution, narrow versus wide, which one would have a better resolution? Same concept with thickness. Okay, so when you have a narrower column, you have less stationary phase in that narrow column. Okay, longer, narrower column, that means you have better resolution. So if you want to compare low pressure liquid chromatography versus high pressure liquid chromatography or high performance liquid chromatography, what are the advantages of the high performance liquid chromatography from what we talked about last time? Say that. Speed. Thank you, Georgino. Yeah, it is speed with high performance liquid chromatography. You have shorter analysis time. You have better resolution because your column is packed under pressure. You don't have very much void between the particles. So you have sharper peaks. You can collect the samples better. That means because you have better resolution, your components are separated better. You don't have like overflowing peak, like a wide peak. So that wide peak could be multiple components, whereas in high performance liquid chromatography, you can achieve better separation and you can collect purer fractions. And then the columns are reusable, they are durable, and whereas in low pressure liquid chromatography, oftentimes you have to empty the column and repack again before you can use it, and sometimes you have to get new media to pack your column. What is gradient elution? Kind of like we talk about it a lot. It's just kind of refreshing your memory so that you know what gradient elution is. Dan, go ahead. Yes. So gradient elution, where we're changing the concentration of the mobile phase over time. So you have gradient elution versus isocratic elution. So what is guard column and what is its function? Chrissy? Exactly. You put it on the front of the column to protect the column from impurity. So it kind of acts as a filter. Even though you extract your components and you filter them, you still have some constituents that might bind to your columns and would reduce the availability of functional groups. So it's guarding the column, it's protecting the column from those impurities. How many solvents and pumps can one use in HPLC? Two, I see two here. Maybe three or four. So you can have up to four really. Sometimes you have one pump and you can have two solvents or three solvents or four solvents, but one pump is pumping them all. We have this system called low pressure mixing. So you can have multiple solvents mixed at low pressure and one pump is pumping. Or you can have one pump per solvent. You can have two pumps, two solvents, four pumps, four solvents. So it depends on your setup. Okay. Where is my, here it is. Okay, so you all seen the lab already, seen the video with the different components. So this is just a reminder. We were talking about the different components of the HPLC last time, and we stopped at the oven, which I believe that was the last piece of component we wanted to talk about, and that's where it's a compartment where you place your column to regulate the temperature of your analysis. Sometimes room temperature is perfectly fine for separation, but sometimes you need to run it at 38 or 40 or 45, so you need the oven. But even if you're running at room temperature, it's best to put it in the oven and place the column in that compartment and put that temperature, which is room temperature, 24. Because if you are in a lab and it's summertime and you have your air condition on, and then the air condition goes off and the temperature goes up and the air condition goes back on. So you have a fluctuation of temperature between possibly 28 and 20 degrees Celsius. That impacts your resolution. Every time your components will come out at a different time because the temperature is changing, so their movement is changing, so you don't have consistent retention time. So it's very much recommended that no matter what the temperature you're running at, control it. Put your columns in the oven and control the temperature. And like I said, having a higher temperature really helps with shortening analysis time, getting sharper peak, enhancing resolution, but definitely you don't want too high of a temperature. You might damage the column. Then there will be, and also with the very fast movement, that you would end up components eluting fast and not separating. So again, you want to find this sweet spot. Okay, now we talked about the different components in terms of pumps, injection port, oven, your column, your guard column, all of that. One of the most important things of HPLC is detection, right? So we need a way to detect our constituents, detect them and quantitate them. So we need detectors. We have so many different detectors that we can use in HPLC. The most common detectors that you would see are UV detectors, UV visible detectors. So these detect components based on chromophores. So chromophores, that means they absorb light, whether in the visible light or whether it is in UV light. So proteins are chromophore, polyphenols are chromophore. So these are examples of what you can utilize. Caffeine is a chromophore. Okay, so in UV detectors, you have different types. And in the lab, we talked about two types. One of them is the variable wavelength detector. So that detector is called variable wavelength detector because you can select the wavelength you want based on your component. What wavelength would be best for your component? What's the lambda max, the wavelength at which you have maximum absorption? So you can select that and have your component be detected that way by absorbing at that particular wavelength. So you have two lamps in a variable wavelength UV visible detector, deuterium for UV and tungsten lamp for visible. So they are the source of light. So in this detector, you need a source of light that the sample or the component will utilize or absorb. You have better sensitivity at lambda max, right? So the wavelength at which you have maximum absorption. So we really need to know the appropriate wavelength to use to enhance our sensitivity and get best quantification possible. If you have sample components that absorb at different wavelengths, so you can adjust the wavelengths over the run, but you have limitations. So for example, the UV variable wavelength detector that we have in our lab, we can change the wavelengths only once during the run. So we can start, let's say, at 220, and then after 20 minutes during the run, we know that a different component is going to come out and has a lambda max of 254. So I can program it to change in 20 minutes to 254. At that time, the second component that I'm interested in will have maximum sensitivity. I have the area at the maximum lambda max. But that is the limitation of this is I can only select two. I can select two that you can simultaneously measure absorption at two wavelengths, or I can program it to select it measures at one wavelength, and then after a certain period of time, measures at a different wavelength. But this has limitations if I don't know what is the lambda max of my components. And that's when another UV vis detector, yes, Holland. Yes. So Holland is asking if I can only change it once, what do I do? What do I do if I have multiple, not just two components at different wavelengths? That brings me to photodiode array detector, because that UV detector has a limitation. This one is going to solve that limitation for me. This one, I can detect multiple components and select for every component, select its own lambda max. So that takes care of this problem here, the photodiode array detector. How it's work, I will briefly talk about it here, but then next week when I start talking about spectroscopy, I'll dive more in the detail into how UV spectroscopy goes. But let's see here. I'm just going to use the board again here. So you have your lamp. My drawing is 4, but this is your lamp. So the light from your lamp is going to, in photodiode array detector, is going to go straight to your sample. Let's say your sample is in a cuvette or is passing through in the flow cell of your HPLC. So here's your sample. And then from there is going to go into a prism or a grating system that's going to diffract the light into multiple lambdas, wavelengths, different wavelengths, x1 through whatever. So what happens here is all of the light is going to hit your samples. The different components in the sample are going to absorb a different wavelength. So the light coming out has reduced wavelength power at specific lambdas. Let's say this is 280, this is 254. So one component would absorb at 254, so you have less of that light at 254 coming out, and another one at 280. You generate a unique spectra that will will hit a series of photodiodes. So what are photodiodes? They're just, each photodiode is going to take the light and convert it to electrical signal. And that converts it to your spectra that you would see. Spectra at different wavelengths, let's say for UVs from 200 to 500 nanometers. And then here you have your absorption. So you will get a spectra that is unique for every component leaving the column at a particular time. So this is, you get absorption over wavelength at any time point. 0.1, let's say every 0.1 seconds, you'll get a spectra of what is coming out. So let's say this is your caffeine here that we measured in the lab. This, let's say, is 272 nanometer. So you have the maximum absorption at this wavelength. Okay, so this is the advantage of a PDA over your variable wavelength detector, where in your variable wavelength detector, you have something else happening. In your variable wavelength detector, you have a different setup. So what you have your lamp, you always have your source of light, whether it's tungsten or deuterium. And then instead of hitting the sample right away, it hits your prism or your grating that would diffract the light into multiple wavelengths, different lambdas. And then you have this whole system is called monochromator. We'll talk about monochromator later. The monochromator will be set to allow a singular or a very narrow band of wavelength going in. That's your variable wavelength detector. So we select the width and the position where one wavelength can go through and then hits your sample. Then everything in the sample that can absorb at 272 will be monitored. So here you have absorption over time. So you only see absorption over time at 272 nanometer. That's all you can observe. So that is peak 1, peak 2, constituent 1, constituent 2. If you change during the run, you change it to different wavelengths. Maybe the constituents here did not show up at 272 at a particular time point, 20 minutes, let's say. You changed it to 220, then you observe another peak here. That did not absorb at 272, but you changed it during the run, then you can see that. So that's the difference. PDA in general is much better than your variable UV detection because it can give you a chromatogram at a selected wavelength. You can tell it, just show me the chromatogram at the selected wavelength, and it can give you the spectra. So if you have a coelution, and that was an example for your caffeine and decaf coffee. So there was the caffeine came out in a very crowded spot in the decaf coffee, and you had like 3-4 peaks coming together. And if we just looked at the retention time, we could have selected one of these 4 peaks randomly. And we wouldn't have known if this peak was contaminated or not pure or pure. With the photodiode array detector, we were able to determine that the peak actually is not pure. Something else was coming out by looking at the spectra. So this is another example of pyrene with an impurity. So your chromatogram shows you one peak. There is a hidden peak inside that you don't see. You just think this is just one peak, but actually there is an impurity coming at the same time that you cannot see it. The PDA allows you to see the chromatogram of the impurity, or not the chromatogram, sorry, the spectra of the impurity and the spectra of pyrene. Then you can say, okay, my peak is not pure. I need to change something, do a two-dimensional chromatography to separate it, or maybe change my mobile phase to separate so that I can get the exact area of my singular component of interest. Other than UV vis, fluorescence is another detector that is, or fluorescent detector is another detector is very common too in analysis. It's also based on ability to absorb light, but not just ability to absorb light, ability to fluoresce. And next week we'll talk about ability to absorb versus ability to fluoresce and what that means in more detail. But that means, what it means is when a component absorbs light, it can fluoresce the light. So you go, when you absorb light, when the component absorbs light, goes to an excited state. And as it goes down back to the ground state, it emits light. But the light emitted is of a longer wavelength, less energy, longer wavelength, because some of the energy is dissipated as heat. Don't worry about all of this, we'll cover it next week. But Fluorescence is meaning the ability to emit light when you go from excited state to ground state. So if you have a component that can fluoresce, it actually have better sensitivity than if it's just measuring absorption. Fluorescence, if the component is able to fluoresce, you get better sensitivity by measuring fluorescence rather than measuring absorption. Some of the components that can fluoresce are vitamins and aflatoxins, for example. And we will give examples of vitamin analysis when we cover vitamin chapter. Refractive index is a very common detector, usually used in carbohydrate analysis. So what is refractive index? It's actually measuring the change in the refractive index as your mobile phase is going through the detector. So you have your mobile phase, you don't have any component, and it has a constant refractive index measurement. As a component coming out, the refractive index is going to change, and it's going to change relative to the concentration. So, but the problem here is that anything that changes in the mobile phase is going to be detected, okay? So, but it's not very sensitive. It has to have a certain concentration. So it's not really sensitive, and it does not differentiate between different components very well. And another limitation is you cannot use a gradient illusion with refractive index. Why? Do you think you cannot use gradient illusion with refractive index? Yes, Sam? Yes, but what else will be impacted? Refractive index, yes, Dan? Exactly. So Dan is saying when you're changing the solvent over time, it's changing the concentration of the solvent over time, you're changing the refractive index as well. So you don't have a constant baseline anymore. So the refractive index is impacted by also if you change the mobile phase concentration. So you can only run isocratic dilution with refractive index. Electrochemical detector, and the most common one nowadays is called pulsed amperometric detector, which is electrochemical detector that measures analytes that can be oxidized or reduced. So as the analyte moves through, you apply a voltage, and there will be a change in current that would be measured based on the reduction or oxidation of that analyte. Or you can measure the change in conductivity of between two electrodes of ions. So if you're measuring inorganic ions or organic acids, for example, you can measure change in conductivity. So either you're changing the change in current based on oxidation or reduction, or you are measuring the change in conductivity. Light scattering detector is called mostly a universal detector. So it can detect anything. So the way it works, so you have your solute in the solvent, and when it gets to the detector, the solvent is evaporated, and what is left is your constituent. And the constituent is going to scatter the light. So hence the light scattering. So the more you have, the more light that will be scattered. And it is universal because it can detect anything. So any non-volatile really, because you want to evaporate the solvent, so you don't want anything volatile. Obviously, compounds will not be detected because they will be volatilized along with the sample. MOS is a unique type of light scattering. It's multi-angle light scattering detector that is commonly used in size exclusion chromatography because it gives you accurate mass, not just quantity, but accurate mass that hits the detector. Another powerful detector is mass spectrometry. So you can link your entire HPLC to a mass spec. So you don't have to have a UV detection or any other form of detection. You have mass spectrometry type of detection, which Gary will talk about Friday and Monday. Last, you have chemical reactions, which really if you have a component that is not a chromophore and you have a UV detector in the lab, but your component is not chromophore. So what you would do is you would react it in a way that you would make it become a component that absorbs light. So take amino acids, for example. Only 3 amino acids absorb light. So you have tryptophan, tyrosine, phenylalanine, and amino acids that absorb light. If you're determining amino acid composition, so you took the protein, you broke it down into amino acids, and then you want to determine the composition of amino acids. Not all of them are going to absorb light. So you react them with OPA, a reagent that will react with each individual amino acid and give you a component that will actually absorb light. Then you can measure them using UV detection or fluorescence detection. So OPA usually is linked to a fluorescence detection, and menhydrin is linked to UV detection. Very commonly used in amino acids composition. You can use these questions to study from. Okay, this is a table 13.1. It's good revision table. So when I put a table up, I'm highlighting a table in your textbook that would be good to study from. So this is giving you an example of the analytes, what mode of separation you would choose, and what would be the method of detection. Later on throughout, after we're done with chromatography and spectroscopy and spectrometry, we're going to start chapters specific for different analytes. So we will be diving more into vitamins, carbohydrates, constituents of concern, and such. So we will be talking about methods of analysis. In the quiz or an exam, I would tell you, okay, what chromatographic technique would you use for such analyte? You will tell me GC, HPLC. You said HPLC, I will tell you what's the mode of separation. You will tell me what's the mode of separation. Then I tell you what's the mode of detection. When I say what's the mode of detection, I want you to tell me what detector you're going to use. Okay, so learn these terms. When I ask mode of separation, you know, reverse phase, normal phase, ion exchange, so on. Method of detection, what detector would you be using to detect your component? Just briefly here, I'm going to go over some example application, but again, when we're done with spectroscopy section next, we will go through analytes. But for example, normal phase are mostly for relatively polar components, such as carbohydrates, pesticides, water-soluble vitamins. Reverse phase is relatively non-polar components that you would analyze fat-soluble vitamins, for example, phenolic compounds, alkaloids like caffeine. There's also something called ion pair reverse phase. This you can separate components that have charge, but what you do is you add an ion that would block that charge or mask that charge. Then the component can react via the relative polarity. Here's just an example of chromatograms of a normal phase and a reverse phase. And it's just examples here, FYI examples. Applications, again, for ion exchange, anything that you can make, that you can have a charge on them. So anything that would potentially carry a charge, you can use ion exchange. Amino acids, definitely. Proteins, definitely. Carbohydrates, you would go, glucose doesn't carry a charge. No, it doesn't carry a charge. Or lactose doesn't carry a charge. But do I use ion exchange? Yes, I make them carry a charge. So they are very weak acids. Sugars are very weak acids. So they won't give you they have OH groups, right? But if you put them in a pH that's very alkaline, they're going to lose their H. So they'll become negatively charged. Then you can use ion exchange. In the chapter of carbohydrates, we'll talk more about that. Size exclusion is often used as a preparatory method of separation. So it's not often used in high performance liquid chromatography, but we can use it in high performance liquid chromatography if I'm only after separation based on molecular weight and I want to determine the molecular weight of my components. Then I would use it in an HPLC manner. Otherwise, we use it as a preparatory separation to separate fractions of proteins based on molecular weight and then collect them, starches, amylose, amylopectin, dietary fiber. So large molecules that I want to separate, large polymers that I want to separate and purify, I use size exclusion chromatography. If I want to identify exact molecular weight and separate them based on molecular weight, I can use high performance liquid chromatography instead of low pressure liquid chromatography. Affinity, it's used for specific application to isolate allergens, to isolate and purify enzymes, to isolate and purify toxins. These are, again, FYI, examples of ion exchange and size exclusion. In size exclusion, these chromatograms, you can see the peaks are not sharp, and that would be mostly coming from low pressure liquid chromatography where your peaks are not really sharp. Okay, so I want to spend what's remaining of the time here on this exercise. It might come in an exam or a quiz, I don't know, but I want you to think with me, and on a piece of paper, you can pull out a piece of paper and put your answers on it, take a picture of it, and I'll ask Laura to have an assignment for an extra credit, a +2 extra credit for everybody that submits it. What's today's Wednesday? We'll have it till Friday, 5 p.m. I'll make sure let Laura know. I don't know if she's online. If not, can you tell her, Brigitte? Okay, thank you. All right, so this is an example here of a reverse phase. Well, I'm not gonna say it. I already answered one of the questions. Anyway, it's a chromatogram of 12 isoflavones extracted, or they're actually standards. You'll be working with isoflavones starting next week in the MS lab. So what you see here, you have 12 different components that were nicely separated. And then at the bottom is the method. How did we separate them? So we started with 17% acetonitrile. That means 83% water, 17% acetonitrile at 0 time. Then we increased the concentration of acetonitrile over 25 minutes up to 25%. And then we kept it constant for 5 minutes. Then we increased it to 30% over 10 minutes. We left it constant at 30% for 10 minutes. And then we went down back to 17% and we equilibrated at 17% for the rest of the run for about 8 minutes. Okay. Is this normal or reversed phase? Okay, I already spelled it out. It's reverse phase, but how would you have guessed it was reverse phase? Let me rephrase that question. How would you have determined it was reversed phase? Yeah, let's see if anybody other than from what you're seeing here, how would you determine its reversed phase? Okay, Steve. Polarity of the starting solution. Yes. Then was that what you were saying? Okay. So we're starting at the beginning here. 17% acetonitrile, 83% water, so it's more polar. And what I'm doing is I'm increasing the acetonitrile, that means I'm reducing the polarity. So this gives you the hint that it is reverse phase. Okay. Which isoflavone is the most polar? You don't have to say its name. You can say the first one or the last one. See? The first one. So in reverse phase chromatography, the first peak or component is the most polar because it will not like to interact much with the with the stationary phase. It would like to interact more with the mobile phase, so it moves fastest than the other. So the first component is the most polar and the last component is the least polar. These are all common questions that I would ask in a quiz or an exam. Okay, this one is good. How would you change the mobile phase so that genistein, which is the last component, would elute sooner, I mean comes out sooner, without changing the relative position of the other compounds? So I don't want to change anything from those compounds that are coming out first. But how can I? So between, let's say, 33 minutes and 48 minutes, there's like 15 minutes here. And this one is coming out that late. How can I make it come out here, let's say? Chrissy.
In your method, it takes the polarity of the metal, decrease the polarity of the metal phase.
Yes. Decrease the polarity more so that I would add acetonitrile at higher concentration. Okay, I forgot that I'm moving away from the computer. I would add I would increase the concentration more. So let's say here, this one, A-gin, is coming out, let's say at 34 minutes. After 34 minutes, after this one is coming out, I can increase it faster or more, maybe 40% acetonitrile. Then this one will come out sooner and my run will be shorter. Okay. What would happen if you keep an isocratic elution mode at 17% acetonitrile? So if I don't do grading, what's going to happen? If I'm running only 17% acetonitrile, what I'm going to see? Anastasia. Longer elution time and wider peaks too. So I might lose this nice separation here. So the separation between these, let's say 3 here and 3 here and 3 here, they're very close to each other. So if I'm just running isocratic, they're going to take a long time. Each peak is going to be wider. They're going to be overlapping. So longer analysis time and loss of resolution of peaks that are close to each other. Okay. What would happen if you keep an isocratic elution mode at 30% acetonitrile? So instead of starting at 17, I started at 30% from the beginning. And then I kept it 30%. And so I get overlapping peaks. Yes, sure, I'll get shorter analysis time, but these two will not separate. They're going to come closer, maybe even in my void volume. We will not separate them from the solvent front. These three will come out as one, these three as one, these three as one. So the analysis time will be shorter, but I lose separation. That's why here you see sometimes I go up. All right, again, I'm walking away. Sometimes I go up and then keep it constant for a while. I kept it constant between 25 and 30 minutes because here I want to make sure that they separate. If I continue increasing, these three might not be separated. So I have initiated some constant concentration to ensure separation of my peaks. Okay. All right, so the very last thing, I have a few minutes. Here is an example where we use high performance liquid chromatography in multiple dimensions. So 3D here. And this is an example of, let me just close out of this chat for a second so that everybody can see. Okay, so this is an example where I want to separate a bioactive peptide. So I want to create a nutraceutical that is made up of a peptide that is specific for a physiological function. So I take my protein, I hydrolyze it to release that peptide, and now I have a mixture of peptide. So I want to separate those and just take the peptide that is bioactive. So how do I do that? I'll start with a mixture of my hydrolysate, so protein hydrolyzed. I have one of those peptides, is the peptide that I want to produce my nutraceutical from. So I use ultrafiltration first step because I want to get rid from all large molecular weight peptides and polypeptides that are left. I'm only looking for those small peptides. So I choose, let's say I'm choosing a 5 kilodalton cutoff. So I filter everything in an ultrafiltration membrane where I get rid of anything bigger than 5 kilodalton, and I take the components that are smaller. So that's my first step. Next, I can do high performance liquid chromatography to separate based on size or based on charge. Peptides, they carry charge and they're different in sizes. But I might have 2 peptides of the same size, or I might have 3 peptides of the same charge but have different size. So I need to separate them using 2 dimensions. So I do charge, and then I follow by size exclusion, or I do size exclusion and then follow by charge, by ion exchange. It doesn't matter which order, but I can do either one first. So you can see here, so I do my first chromatogram. I get my first chromatogram, size exclusion or ion exchange. And then what I do is I collect each fraction and I run my assay. The assay of bioactivity assay, whatever it is, antioxidant activity, for instance. I take the fraction that has the highest activity. I take this fraction of the highest activity and subject it to size exclusion chromatography or ion exchange, whatever it is. Then I separate them more. I take each fraction and then I determine which one has the highest bioactivity. From there, I use maybe reverse phase chromatography to separate them further. So I run, because they might have same size, but you have multiple peptides. So you run them on reverse phase, then they will interact with the column differently. Again, and you do the measurements to see which fraction has the highest activity. I take that fraction, and to identify it further, now it's a pure fraction, I run it on LCMS, or I do N-terminal or C-terminal sequencing, which is advanced mode of analysis to determine the peptide sequence. Then I would determine the sequence and I confirm its bioactivity. So this is an example where you can do multi-dimensional HPLC to purify a particular component. We're done with chromatography. We can start mass spectrometry this Friday. Okay, see you all on Friday.
I have a question.
Is that Carly? I can barely hear you. There's something off with your audio. It's kind of like a robot. Oh, can you